I'm going to present evidence for evolution by natural selection. And I want to introduce two concepts. One is the macroevolution. Macroevolution happens at the macro scale level. That is the comparison among species. And we're going to go through this example, evolution of the forelimbs. The other concept is microevolution. That is at the macro scale level. And we're going to go through some series of examples that are actually very important or relevant for humans. That's the evolution of drug resistance. And we have three different examples. For the macroevolution, I want you to think about this. All of these examples here, humans, cats, whales, and bats, have something in common. So they have structures that at first glance, they look different, but they actually share a lot of history. These are homologous structures, and they share a common origin or are similar anatomically, right? So if you look at these colors, they are bones that are found in all of these four examples. So let's take a look at this one. This is the radius. And the radius in orange is present in humans, in cats, in whales, in bats. So through natural selection, these different species, remember macroevolution happens at the species level. So through a series of generations, through a long period of time, natural selection has shaped these homologous structures to look different. And humans have become adapted to have this opposable thumb, for example. And now we can grab our cup of coffee, we can easily write, and we can do a lot of things because we have evolved these adaptations. How about cats? So cats have these extremities or these forelimbs that allow them to run faster. Whales have these homologous structures that they use to swim, and bats have these structures that share the same origin, but they use it for a different purpose. They use them to fly. So you see, they share the same common origin, and they might be different. So those are homologous structures. Now, the evolution of drug resistance in bacteria, this is one example of natural selection, evolution by natural selection at the micro scale level. Microevolution is happening here. So in here we have one strain of methicillin-resistant Staphylococcus aureus, MRSA, which is a very dangerous pathogen. It's a eating flesh type of bacteria. And there are different type of strains. Oh yeah, I have a helper here. This is MRSA, this is bug. This bacteria, thank you. This bacteria, actually, it's a different strain. So it has a mutation compared to another. Let's imagine that this is the normal bacteria, right? So this bacteria is harmless. This one is very harmful. This one, MRSA, has some genes that are resistant to penicillin. And then you get in troubles if you get it very active in your skin, because there is no antibiotic currently that can get rid of them. Or, well, there are some strains that are more difficult than others. So Staphylococcus aureus, you can find it in your body. We have it commonly in our body. The problem is when we expose our flesh to this bacteria, and the bigger problem is when the strain has resistance to penicillin or methicillin. These Staphylococcus aureus become resistant to penicillin in 1945 and then to methicillin in 1961. So it can be really bad, right? So some people can lose some parts of their bodies if they don't get treated appropriately. if we have these two bacteria, they are the same species as Staphylococcus aureus, but one strain is going to be susceptible to antibiotics, and the other one is going to be resistant to antibiotics. Then if selection acts, this means the antibiotic acts and kills this bacteria, we're going to get only this one, right? So the drug is not creating this bacteria, it's just selecting for it. So mutation created this new strain. Another example is the evolution of drug resistance in HIV patients. In this graph, you have the x-axis here, and you have weeks, so from 0 to 12 weeks. In the y-axis, we have percent of HIV resistant to 3-TC. That's a drug. That's antiretroviral medication. So that means that you're going to kill this virus. And it's used in patients with this disease. What you can see is that the resistance, the virus, so this is measuring 3 different patients and how rapidly the virus gains resistance. And that is because a virus is a very small thing, and we still debate if viruses are alive or not in nature. There's some people saying that they are alive, but well, let's call them things for now. These things actually replicate really fast, and then in few weeks you can accumulate new mutations. So this is the result of very rapid selection acting upon these strains of virus. This is the last example of microevolution, evolution by natural selection, and it's the resistance to DTT, the dichlorodiphenyl trichloroethane. And this was a chemical that Paul Hermann Müller discovered and used as an insecticide, and he won the Nobel Prize for his discovery in 1939. However, later studies found that mosquitoes actually gain resistance to DTT really fast, and we can see in this table the months. In 0 months, in less than 1 month, 4% of the mosquitoes gained resistance. So applying more DTT was not killing the mosquitoes. And then later, if you keep killing mosquitoes with DTT, some of them will die, but a lot of them will be more resistant because you have in the population some mosquitoes that were already resistant. And then you increase the numbers, and as you keep going, most of your population that were left of the mosquitoes have gained resistance. So if you keep going this, all of your mosquitoes are going to have resistance, and you're not going to be able to control the mosquitoes with DTT. So that's why nowadays there are some very strong approaches to disappear the mosquitoes from Earth with genetic engineering. And that's the same idea that if you keep using antibiotics and the bacteria keep gaining resistance to the antibiotics later, we won't be able to control bacteria with antibiotics. And actually, we are facing that crisis right now. For that, it's very important that we start investigating other alternatives, such as the use of bacteriophages. Do you remember the experiment of Martha Chase and Hershey? Well, they use this bacteriophage, and bacteriophage are viruses that kill bacteria. So if we use those, it's more a natural approach to kill bacteria, and that is a very interesting topic that I will encourage you to follow if you are interested in this area. Okay, so this is how we go from 4% to 77%. And if you were curious about what DTT does to the mosquito, it's actually very aggressive. DTT opens some channels in the neurons and opens these sodium ion channels and cause them to fire spontaneously. So I imagine them like having some scissors, and that leads to spasms and eventually death. So if some mosquitoes have mutations in their sodium channel gene that are resistant to DTT, they might become resistant to this insecticide.